Hello students, how are you people? Hope you're all doing good. Once again, welcome back to our biology class. Myself, Dr. Raghavendra here. Fine, in the earlier class, we studied about how the enzymes catalyze various kind of reactions. Yes. So reactions can be physical, reactions can be chemical. Under chemical reactions, it can be catalyzed, uncatalyzed, organic, inorganic, exothermic, endothermic. Yes, we have discussed all about those reactions. And the enzymes also known to increase the rate of reactions that we have discussed with a simple example as such a formation of carbonic acid in the presence of enzyme carbonic anhydrase. 6 lakh molecules will form, but in the absence of this enzyme, only 200 molecules are formed per hour, whereas 6 lakh molecules formed per second means 10 millions of times rate of reaction is accelerated with the presence of the enzyme. This we have studied. And also we studied about the activation energy. Yes, amount of energy required in the presence of the enzyme to convert a substrate into product is very less. In the absence of enzyme, yes, activation energy requirement will increase very high, and it will be, yes, going to form the same amount of a product within the same given time, but energy utilized is very high. That's why to reduce the energy, to increase the productivity, enzymes are must and should. So let us understand now nature of enzyme action, how actually the enzyme action will be carried out. So when we talk about the nature of enzyme action, nature of enzyme action. So before we go into the detail of the nature of enzyme action, let us understand now the structure of mainly the enzyme. Yes, it is just a diagrammatic presentation what I am going to give you, not a detailed chemical structure I am going to give. Yes, when I talk about the structure of a basic structure of an enzyme, what it represented. Enzymes are, yes, as we have discussed, they are basically, by chemically, they are the proteins. So I am going to draw here. It is an enzyme. Yes. So this enzyme, it's a structure, what we call it as basically. This is all the protein. So this protein part is called as apoenzyme. Apoenzyme, which is, we say, it is the protein part. So it has, what we can say, that another part. So we can say it can be a prosthetic group. Okay, so we call it as mainly, it can be the cofactor. Cofactor are those important non-protein parts which are attached to the enzyme. They increase the efficiency or catalytic activity of the enzyme. Cofactor, non-protein part. Non-protein. So cofactor sometimes referred as coenzyme also. And then this is what we can say, the binding site, substrate binding site we call it as. So this is mainly the substrate binding site. This substrate binding site sometimes referred as active site. Also. Well, so substrate comes over here. Yes, substrate has a specific structural conformation. Yes, it come over here and it bind with this. So this is what we say, the substrate is, they get fixed well. So this is the substrate, right? So the substrate fits very well in the active site only, nowhere it go attach. So this is what we talk about, mainly the substrate as such. Now the nature of enzyme action as such, the conformational, what we can say, structural analogy will be there. Proper or particular substrate will bind the particular kind of enzyme only. So enzyme substrate structures are very specific. So here what you are observing, substrate will come and bind with the enzyme at a particular active site, presence of cofactor as well as the protein part of the enzyme called as mainly apoenzyme. So the cofactor and mainly the apoenzyme together, it is called as a holoenzyme. holoenzyme. This we need to understand very well. So, thus what we have seen the enzyme mainly when we talk about the nature of enzyme action as such. What are the important thing mainly they reveal? So, in the structure of enzyme we observe basically there is an active site or substrate binding site. We also observe mainly there is what we can say mainly there is a cofactor, presence of cofactor increases the efficiency of an enzyme. And then yes, we also observe mainly the substrate has a structural conformation, so its structure is perfectly matches with the active site, so it can bind well. So that's what we call it as enzymes. So mainly we can say that enzymes act on specific substrate and catalyze particular reaction. So this is about mainly the enzyme. So as such, how the enzyme as well as the substrate, initially let me tell you one simple yes equation. E refers to enzyme plus substrate, then they form interconvertible ES complex. Yes, this substrate pass through the transition state, then it become EP complex, enzyme product complex. Further, what happens? The enzyme and product get detached. So now this enzyme is set free. It is set free. So now this enzyme now ready to act again to go and bind with the new substrate. This is what we say mainly the mechanism as such. So when you look into this, what will happen? Let me tell you, as such we have seen. This is the enzyme. Okay. Yes, enzyme has its cofactor attached here. So we say plus, mainly the substrate is coming. Substrate will be like this. Okay. So this is the enzyme plus substrate. Further what happens here? I hope you are able to draw this. So cofactor is here. Now it become mainly ES complex. Okay. So it become ES complex. Further transition state. So here what will happen mainly? The enzyme remains as it is. It doesn't undergo much of the changes because it is a catalyst. So now what happens? The product is formed here. So now the product is formed. So we say EP complex. Further, the enzyme get detached or separated. It's having its own cofactor plus we say that its product. Yes. Now this enzyme plus the product. Enzyme is now free once again to go and form enzyme substrate complex. This is what we are observing, the nature of enzyme action. So as to say that the nature of enzyme action reveals 4 important steps. Number 1, enzyme bind with the substrate, specific substrate. Okay. Number 2, the substrate attached bring structural changes. So specific substrate, the enzyme binds to specific substrate. So substrate attached, we can say that to active site, bring structural changes in enzyme. So binding becomes much more tight. At this time, yes, binding become tight. Number 4, yes, substrate undergo transition state. That means chemical bond broken and reformed. Bonds broken and rearranged. Then number 5, substrate. Now we call it as enzyme substrate complex. Yes, binding becomes tight. Yes, here it is called as ES complex. Now substrate is getting converted chemically as well as physically. You can say that substrate converted into product. Thus EP complex. Now product is released. The free enzymes, okay, the enzyme set free. Set free to bind with a new substrate. To bind with new substrate and catalyze some more biochemical reaction. This is what we observe basically. Yes, here what you are observing mainly the nature of enzyme action, the structure of enzyme, what we have seen mainly the enzyme bind with the specific substrate and then ES complex is formed, EP complex is formed, then enzyme set free by releasing the product. So this free enzyme is once again ready to bind with the new substrate. So every enzyme has its own specific what you can say substrate. All the enzyme cannot react on each and every substrate. So they have specific substrate to act upon. This is what we say the enzyme substrate, yes, activity or nature of enzyme action. Well, so we say that basically enzymes they have particular the substrate binding site we call as a active site, and they do have the cofactor or we call them as a coenzyme plus apoenzyme, the protein part. So cofactor can be non-protein also, it can be metal also, right? It can be vitamin also, fine. So they are all called as prosthetic groups, okay? So that's what we say that mainly the enzyme catalyzes the reaction and reduces the required amount of energy, convert the substrate into product. So this is all about the nature of enzyme action. Well, so I hope you have noted down all these points which are very very important and essential. Fine. This is all what we say mainly the catalytic activity or catalytic cycle done by the presence of enzyme. Fine. So the next concept, yes, what we are going to discuss is all about mainly the factors influencing the enzyme activity. Let us understand that also. Well, let us go ahead. How the enzymes mainly altered by the different kind of factors. So after that we will discuss about classification of enzymes and different types of cofactors also we discuss. So this is about what we have studied, nature of enzyme action. Fine. Enzymes are basically, yes, sensitive biomolecules. So why they are sensitive? Because they are proteins, and proteins are sensitive to the fluctuation in the pH, fluctuation in the temperature, the concentration of substrate also important to catalyze the reaction. Okay, so all these, yes, we are going to discuss one by one. What we call it as, we study these under the title factors influencing the enzyme activities. So each enzyme has its own range of pH or temperature. So let us understand the factors influencing enzyme catalytic activities. Okay, fine. So enzymes being proteins, what they are made up of? Enzymes are basically made up of amino acids, right? So enzymes being proteins, and they are mainly made up of amino acids, and the proteins are highly sensitive to the pH as well as temperature. So thus the factors influencing the enzyme activity, one is we can say that temperature. Number 2, it is the pH. Number 3, it can be substrate concentration. And number 4, we say that yes, substrate concentration. Other than that, one more important, inhibitors. Inhibitors also known to, yes, mainly affect the enzyme activity. So we need to understand all these one by one. Let me tell you here, when we talk about temperature, most of the enzymes, so first let us consider temperature. Most of the enzymes, okay, act at body temperature. That is 37 degrees centigrade. Okay, so enzymes being what we can say that low temperature, what it does, temporarily inactivates or reduces enzyme activity. That's why when we keep the food in the fridge, no much enzyme activity, no spoilage of the food. Even in a cold condition, if our body temperature fall down, there is no proper metabolic activity happening inside our body, and we feel laziness or dizziness, right? Lethargic condition. So at low temperature, most of the enzymes remain inactive, right? Under a freezing condition, enzymes will not work out. So the enzymes at high temperature get damaged or denatured and become inactive. Okay, so thus enzymes activity, yes, increases only at optimum range. High temperature causes the denaturing or damage of the enzymes. Low temperature make it very much inactive. We can say that most of the body enzymes are very active at a range of body temperature of 37 degrees centigrade, and then there is one more important point we need to know what it is. So, when you talk about the increase or decrease in 10 degrees centigrade of temperature, so we can say that every plus or minus 10 degrees centigrade can increase, okay, increase or double the rate of reaction, double the rate of enzyme activity, or the enzyme action reduced to half. Okay, so every plus or minus 10 degree temperature, whether the temperature is favorable or unfavorable, it depends on the metabolic pathways. Under such situation, what happens? There may be a favorable temperature range as the enzyme is thermolabile, it is heat resisting also. Rise in 10 degrees centigrade, yes, there may be suddenly increase in the enzyme activity, or there may be a chance that the increase in temperature also damage the enzyme, and then enzyme activity may go or fall down to half of its actual efficiency or activity. This is what we need to understand. Enzymes are mainly heat sensitive. They are basically made up of amino acid. They are the proteins, and they cannot tolerate the temperature beyond the range of optimum conditions. So similarly with the pH also, let me tell you here, second important thing is pH. The first one what we discussed, it's all about the temperature, pH. So beyond optimum range, the change in pH can affect the enzyme activity. So efficiency of enzyme may fall down. So there are the enzymes which are active at acidic condition. Let me mention here the pH range as such 2.0, and then the enzymes such as pepsin in stomach remain active. Then at a pH 8.0, the enzyme trypsin, in intestine remain active. So thus there is a different what we can say range of temperature as well as pH which favors their activity. To say here mainly the enzyme activity and it may be the pH or the temperature. Okay, so enzyme catalytic activity increase and again start declining. So this is what we can say the optimum range of pH or temperature that favors mainly increase in the enzyme activity. So thus this peak enzyme activity attains maxima only at optimum range, beyond the optimum range enzyme catalytic activity will fall down or reduced because of change or fluctuation. So here in fact it should be what I can say here, it should be the enzyme activity and it should be the pH or temperature over here. So pH or temperature. Here it should be the enzyme activity. Well, this is what we talk all about, mainly the enzyme activity at a given particular range of temperature, or at a particular, what we can say, the pH. So the concentration of substrate and enzyme activity can be well studied by one more, yes, what it called as a Km value we need to understand. Let us understand mainly substrate concentration. Yes, the more we keep on adding the substrate, so here let us understand that also. Substrate, increase in the substrate concentration only up to certain level can increase the enzyme activity. Substrate concentration increase in substrate concentration up to some level increases enzyme activity. Enzyme activity. So once all enzyme bind with substrate, once all the enzyme bind with the substrate, addition of extra substrate cannot alter, cannot increases enzyme activity, because all the enzymes are already bind with the substrate. There is no more free enzyme available to bind. So addition of extra enzyme, the substrate cannot alter the rate of reaction, we can say. So a simple graph is very, very essential to understand this. So how this substrate concentration and we can say that enzyme rate of reaction or velocity of the reaction rate is correlated. Let us understand this. So when we say that here the y-axis we call it as the velocity or the rate of reaction. So it is V max here, the maximum, and then here mainly what we say the substrate concentration. Yes. So what happens as we keep on adding the substrate, the enzyme activity keep increasing, but it become a rectangular hyperbola. Once it attains the Vmax value, we call it as a rectangular hyperbola, and it has reached Vmax value, and half of this we call it as Vmax by 2, and then yes, it intercept or it cut the curve here, and this value or concentration of substrate is called as a Km value. So the Km value is the amount of substrate concentration to attain the half of the velocity. Okay, so thus we can say that substrate concentration Km value is the substrate concentration to attain Vmax by 2. Thus we can say that the affinity between the substrate and enzyme is well measured by the Km value. So higher the what we can say affinity, lesser the Km value. Okay, so thus we can conclude that so the correlation between the velocity or we can say that the enzyme activity, so higher higher the Vmax, lesser the Km value. So thus the Km value is an indicator of the affinity between the substrate and the enzyme. It is the measure of substrate and enzyme affinity. Well, so this is all what we have studied. It is also yes denoted as the Michaelis-Menten constant Km value. Km value differs from one enzyme to the another enzyme. Km value are very much distinct for each and every enzyme with its particular affinity for the particular substrate. This is all yes what we have discussed in the today's class. So in the next class yes we are going to discuss about mainly how the inhibitors affect the enzyme activity as well as the classification of enzymes and applications of enzymes also we'll study in the next class. Till then, take care, study well. Thank you very much.